Hi, everyone. My name is Zane Thornburg. I'm a graduate student in Zane Schulten's lab at the University of Illinois. And today I'm going to talk to you about our efforts of simulating a whole cell model of JCVI Syn3A over complete cell cycles. And a lot of the results I'm talking about today came from our cell paper that came out with JCVI and James Senz's lab back in January called Fundamental Behaviors Emerge from Simulations of a Living Minimal Cell. So I don't need to introduce the system to the crowd, I don't think, but unfortunately, I don't have time in 10 minutes to fully describe to you how we can simulate 7,200 chemical reactions inside of a cell. But to give you a general overview, it takes a lot of data to create an initial cell state. So before we evolve a system of reactions through time, we need to come up with an initial condition for the cell. So to come up with this, we survey any number of omics data that we can come up with, hopefully from bacteria, but we take whatever we can get. And then to do three-dimensional models, we need information such as ribosome coordinates and membrane boundaries from cryo-electron tomograms that we got from Elizabeth Villa's lab at UCSD. And then to evolve our system through time, we need to write down our reaction networks. So I'll briefly introduce those in the next few slides. So this top left panel here, we can introduce sets of reactions like metabolism, transport, genetic information processes, cell division. And then we evolve our system through time using methods that I will describe soon, but we use hybrid stochastic deterministic methods, where some reaction events are random and some are deterministic. And then as we simulate the cells through time, we get complete cell states where we get counts of all the molecules. So for example, messenger counts, we can calculate ATP costs, and we can get population-wide distributions, for example, cell doubling time. And so our stochastic method that we use to evolve the cell through time is known as the reaction diffusion master equation. Here I'm going to talk about the hybrid version because we also include ODEs for metabolism. But we talk about the time evolution of the probability to be in a state X, where X contains the coordinates and chemical identities for each particle in our system. And simply, we can either, we discretize the three-dimensional space of the cell onto a lattice, and you can, at any one time step, you can either diffuse from one site to another, or once you are within one lattice site, you assume that the system is well stirred and you can undergo chemical reactions. And then additionally, if you remove spatial heterogeneity from your simulations, I'm going to talk about some simulations today where we do that, and then the whole cell is a well-stirred system, and we call those chemical master equation simulations. And so just to tell you what I mean by genetic information processing reactions, I'm simply talking about DNA replication, transcription of RNAs, translation of proteins, and degradation of messenger RNAs. And we do this explicitly for each of the 493 genes in JCVI Syn3A's genome. And so don't squint your eyes at your screen too much at this. This is just to make you appreciate that the metabolic map of JCVI Syn3A can fit on one page. So to work through the metabolism, we divided it among coworkers. So my coworker David worked on the lipid metabolism. Troy did nucleotides. I did cofactors and glycolysis. And we spent a lot of time to simulate these deterministically, collecting kinetic parameters and thermodynamically validating our kinetic model of this metabolic map. And so now that you know the two different methods, how do we communicate between stochastic and deterministic reactions? So my coworker David actually wrote a paper back in 2018 where he introduced such a method, where here we have a chemical master equation simulation communicating with ODEs, and you will simulate 1 second of stochastic. So that's the time scale we used. You will simulate 1 second of your stochastic simulation. You pause your simulation. You communicate necessary information to your system of ordinary differential equations. You simulate 1 second of your ODE reactions, and then you communicate information back to your stochastic reactions. So just I have some examples here on the left, but my best example is probably when we're dealing with proteins. The metabolic reactions, all of their rate laws are dependent on the concentrations of the enzymes. So as you stochastically make proteins, you need to communicate those enzyme counts to your metabolic reactions for their overall rates. And so here is a spatial model where we simulated 20 minutes of the cell cycle, the first 20 minutes where we didn't assume any growth yet, no change in DNA, and we fixed the ribosomes. And I've hidden the 77,000 proteins from the proteomics just to make you appreciate that anytime you see a ribosome go from yellow to purple, that means a messenger RNA in our simulation has diffused there in three-dimensional space, bound to the ribosome, and is now being read one messenger at a time. Similarly, the degradasomes, the bacterial mRNA degradation complex, has messengers diffused to those, and those complexes are only in the membrane. And then we also can have the RNA polymerases diffused to genes and actively read the genes one base pair at a time. And so from these shorter simulations where we did the spatial model, we can get some genome-wide statistics. So for example, we can learn the active number of each complexes. So for example, the active RNA polymerases, we have about 180 RNA polymerases in our system, and we estimate that roughly 65 of them will be reading a gene at any one time. And this is actually in the agreement with the range reported by Bremer and Dennis a while ago. And then we can also get some really interesting distributions, my favorite of which is the distribution of mRNA half-lives, where for each of the 452 protein coding genes in the system, I calculated the average half-life over a population of 10 spatial simulations. And so the average and median are actually in reasonable agreement with values that were measured in Mycoplasma gallisepticum. And then this long tail out to 15 has even been observed in Bacillus subtilis. So some of these agreements with previously reported measurements are giving us a lot of confidence in our simulations. And I just want to emphasize here why we use the word emerged. We did not tell the simulations at any point what the half-life was of any of these messenger RNAs. These came out of getting our kinetics right, having all these length-dependent kinetics, getting our diffusion constants right, and really doing our due diligence with all of these other factors and getting these bulk properties predicted. So now to move on, we used those to parameterize a well-stirred model. So where we are now assuming that the whole cell is a well-stirred system, and we simulated that for the complete cell cycle of SYN3A, so out to 2 hours. And so what I'm first going to highlight is that we calculate the ATP cost of every reaction explicitly. And so here we have the orange is the ATP that we generate, and the black is the total ATP cost of the cell as a function of time. And you can see that in general, we're making more ATP than we're using. So in general, we're actually continuing to build our pool of ATP rather than running out of ATP. But a more easy way to look at this, to look at the distribution of costs, is actually to break this down as the fraction of the total ATP cost. So we're just going to set the total ATP cost to 1, and then break down what the fraction is for each component building up to that. And so first I'm going to point out that we can have multiple replication events per cell cycle. So some cells only have one replication event, highlighted by that the ATP cost is only present for one period of time. Or some cells in our simulation, we allow them to undergo multiple replication events. And you can see two different ranges where early in the cell cycle we have the first replication event and another round starts later in the cell cycle. Perhaps one of the more fascinating predictions from our simulations is the total cost of activated transport in JCVI SYN3A. So because of the reduced nature of the minimal cell, it has very few synthetic pathways left, as we all know. So it actually has to take up a lot from the medium. It's this very rich medium that has to take up a lot of nutrients from. So it turns out in our simulations where we're counting every ATP very rigorously, that about 20% of the overall ATP cost of JCVI SYN3A is just invested into taking up nutrients from the environment. And so as I mentioned earlier, we can have multiple replication events. And so one of our other good predictions was Andras Cook, while he was at JCVI, performed some qPCR experiments measuring the origin to terminus ratio for JCVI SYN3A. And he got a value of about 3.4 origins per termini, which strongly suggests multiple replication events per cell cycle. And we have some theta structures of the DNA here on the left, where I'm just showing you diagrams of what the different ratios would look like. So on average, Andras's experiments would predict that it's some average between these two states, typically the 3-to-1 of origins determine I and the 4-to-1 origins determine I. And you can see from our simulations that we're a little bit low, but our uncertainty bars are a bit larger, so we're still within the range predicted by the experiment. And now to start closing it all off, we're going to get toward the end of the cell cycle. So as to grow the cell, as I mentioned earlier, we have lipid metabolism and we have translation of all 452 proteins. So as the components are synthesized, we take the surface area contribution of each lipid and each membrane protein to dynamically calculate the surface area of the cell. So we know what the surface area is because of the components, and we currently have made a simple assumption about the division time, but we predict that the volume doubling time is roughly around 60 minutes based on this growth rate. And then if you look at the surface area curve, if you assume that the time to double the cell is the time to make 2 new membranes to close off the cell, we get only a 5-minute difference between the experimentally measured doubling time and our predicted surface area doubling time. But now, I said we made an assumption here about what the volume change is like. So now we need to move to a spatial model and try to learn a little bit about how the cell divides, as we heard from James's talk earlier. So in collaboration with TJ Haas lab at Johns Hopkins, we have some fluorescent images of dividing Syn3A. And so all I'm trying to point out in these is if you look at the pair of cells in the center, we are fairly confident that these are a pair of a cell that is currently undergoing division because of the connectivity of the membrane intensity. And so we are just trying to show here that the cell divides roughly symmetric. And we know from James's talk that Syn3A has the necessary mechanical proteins to divide the cell. And so our first assumption is just going to be that the cell, to divide the cell with simple geometry, since we know the surface area as a function of time. So we know this property of the cell because of our components, so we're just going to use that at the moment. And so if we do this, we get this nice 3-dimensional model here where the ribosomes are doubling according to the number predicted from the tomograms, number of membrane proteins are roughly doubling, and that the cell is dividing with a surface area growth rate corresponding to the insertion of lipids and membrane proteins. And so what'll be interesting here is that with these stochastic spatial simulations, we are going to see that these macromolecules will partition themselves between the two cells heterogeneously. So it's not going to be perfect division because of the stochastic nature of diffusing between the two cells. So it turns out here at the end that there's actually about 550 ribosomes in the cell on the left and about 490 ribosomes in the cell on, or 550 on the right, 490 on the left. So we get these really interesting distributions now where we're going to be able to predict how the distribution of macromolecules occurs cell to cell. So in conclusion, while a lot of experimental data is used to construct these whole cell models, a lot of these behaviors emerge from the simulations and are not imposed. So we didn't tell it the half-lives, we didn't tell the simulation what the doubling time was, but we got these nice predictions out by getting our kinetics, doing due diligence with our kinetics and obeying thermodynamics. The communication in our hybrid simulations allows the cells to respond to intra and extracellular conditions that a real cell would experience. And if you check out our paper, the multiple DNA replication events actually turn out to be really essential to maintaining and controlling metabolite concentrations over the cell cycle. So I'd like to thank all of my coworkers. I'd like to thank the JCVI Minimal Cell team for collaborating with us and hosting this wonderful conference for us today. And too many other collaborators to name, but thank you everyone that is involved.
Zane, outstanding as usual.
Thank you, John.
Where do you think your model is weakest in terms of replicating something to do with fundamental cell biology?
So I wouldn't say this is a weak point, but this is a place that's in development. Is we are currently working on developing this division model to be a bit more physical. So as I said, we're currently assuming us making some geometric assumptions at the moment, just to begin to programmatically make some of these algorithms even possible to even simulate such a process. But I'd certainly say one of my biggest places where I would want it to be a bit more physically and biochemically realistic would be right in the division area.
Yeah, so if we could, David's paper listed the genes that David Bianchi's recent paper listed the genes that he thought were critical to cell division that are in SYN3A. And so if your model could take into account, instead of just those 3 proteins, perhaps the whole system, that would be really terrific. But I don't think we're quite there yet.
Yeah. His paper was certainly very helpful in determining what else we need to worry about.
And then, a number of the folks who are listening today work with a different near minimal cell system, Mycoplasma pneumoniae. And in pneumoniae, I can imagine how the genome might be pulled into the 2 daughter cells by the tip structure. I'm not really sure, but because I think it is known that the genome is attached to the tip organelle. But
indeed,
in this,
oh, sorry, John, go ahead.
But in our organism, either there is a protein that helps sequester the 2 daughter genomes into the 2 daughter cells, or it's a stochastic process. And any thoughts about how that might, in your model, is it stochastic, or is the genome being actively sequestered?
So in the 3D model, that is something that Ben is currently working on. Benjamin Gilbert, who's going to speak, I think, next. But I agree. Ben and I have both dreamed of having something known like how pneumonia has a pretty well-known mechanism of attachment. That's the kind of thing we dream of. To just knowing how it works is step 1. But currently, it's essentially going to be somewhat of a stochastic process. For example, in our well, I will say this, one thing that we do know from our well-stored model is we think that some cells might even not complete 1 replication cycle during its cell cycle. So you could even possibly get a division event occur where 1 cell doesn't even get a daughter chromosome because replication hasn't even completed in 1 cell cycle.
Jörg Stulke is on the line. Jörg, do you have any thoughts about, is there a minimal cell protein that is involved in chromosome segregation that we just don't understand?
I think there's something like SMC, right? Isn't it?
Yes,
yeah,
certainly.
We do have SMC.
Hi, this is Zane. In fact, Benjamin Gilbert will talk about that, because all the beautiful work that's being done in Europe by Case Decker and John Marco over here has shown that this SMC can really contribute to the chromosome segregation. What we're looking for, Jörg, is any validation of an attachment organelle, similar to pneumonia. We have some possible candidates, and there's still some unknowns here, but we keep looking at your SynWiki, hoping that you'll report something soon. Otherwise, we'll just assume a few scenarios, being the theoreticians that we are.
Okay, yeah, I think we have to wait.
Yeah, Ben's definitely going to talk a little bit more in detail about this, so I think we should leave that to him.